Good afternoon to you all, ladies and gentlemen. It is a great pleasure for me as the Honorary Secretary of the Nutrition Society to chair this third of our awards at today's very special awards conference. And I will be chairing the Sir David Cuthbertson Award, which is of course in recognition of the wonderful clinical scientist, Sir David Cuthbertson, which this award has been since 1990, so it's a bit special. It's in its 30th year, and it is awarded to either scientists or clinicians who are at an earlier stage of their career, and in particular for their research into either clinical nutrition and or metabolism that has then subsequently led to providing evidence for clinical practice. Now, there is no more better awardee than that which we have in our 2020 Sir David Cuthbertson awardee, which is Sir Dr. Richard Skipworth. Dr. Skipworth is a consultant general and upper GI surgeon at the Royal Infirmary of Edinburgh. He is also an honorary reader in surgery at the University of Edinburgh, so he's an extremely busy man. Awards are not new to him. In 2012, he won the Syme Medal from the Royal College of Surgeons in Edinburgh. And in 2011, he was awarded his Doctorate of Medicine with distinction from the University of Edinburgh for his research in cancer cachexia. And he now holds the post of NHS Research Scotland clinician and leads a very strong world-renowned research group that is particularly interested in cachexia, nutrition status and body composition in cancer patients. Richard is a board member of the Cancer Cachexia Society as well as many other societies, including ESPN. And so really it is with great pleasure and great honour, Richard, to welcome you to give your talk this afternoon from the Cuthbertson Award. So please, over to you, Richard.
Thank you very much. Ladies and gentlemen, I'd like to extend my sincere thanks to the Nutrition Society for the Cuthbertson Award, and thank you to Professor Lanham New. It is a great honour, and it's nice as an Edinburgh-based researcher to be affiliated to such a great Scottish scientist as Sir David Cuthbertson. My talk is entitled Muscle and Fat Wasting in Cachexia, the Metabolic Response to Cancer. These are my declarations. So what I'd like to do over the next half an hour or so is summarise approximately the last 15 years of cancer cachexia research that we've done within our unit. And within that, we've really focused on a kind of bench to bedside approach with an emphasis on human translational research, predominantly taking tissue samples from cancer patients undergoing surgery. I'm going to talk a little bit about the pathophysiology of cancer cachexia and particularly the interaction between the host and the tumour and how this appears to drive skeletal muscle wasting. I'm going to talk about the identification of novel therapeutic targets for treatment of the disease, and also in particular early biomarkers. And this is an area that we're particularly interested in, because what we'd like to do is be able to identify patients prior to the development of severe weight loss, so that we can institute prophylactic therapy before they've deteriorated. I'm going to talk about the importance of multimodal therapies as a system of treating cancer cachexia. And I'm going to talk about some of the challenges that we've had in clinical trial design for cancer cachexia, in particular those surrounding appropriate patient selection and inclusion criteria, and those regarding the selection of appropriate outcome measures. So what is cachexia? Well, the word itself originates from the Greek words kakos and hexis, which essentially means bad condition. Hippocrates gave one of the first descriptions back in the 4th century BC, and he very famously said, the flesh is consumed and becomes water, the abdomen fills with water, the feet and the legs swell, The shoulders, clavicles, chest and thighs melt away. This illness is fatal. If we look at the research timeline for this condition, well, it was really first identified in the 1940s. Clinicians became interested in Simmons cachexia, which was really muscle wasting related to destruction of the anterior pituitary glands, largely through processes such as tuberculosis and malaria. Jail cachexia was a kind of wasting associated with prison time, which often happened in patients who had scrofular or peripheral tuberculosis. It wasn't until the 50s that people really began concentrating on cancer cachexia along with anorexia nervosa. And over the next 10, 20 years, it became noted that these sort of muscle wasting conditions were predominant in other disease processes as well, such as heart failure, cirrhosis, and burns. The 1960s, we saw the emergence of tumor-bearing animals as a model for investigation. We also saw some of the first investigations of the histopathology of cachectic muscle. In the 70s, attention really focused on the treatment of patients with this disease. And there were some of the fledgling studies of the use of TPN and the investigation of the tumor-host interaction. In 1980, Device published his very seminal work on patients presenting to palliative chemotherapy and how weight loss influenced their outcome. He noticed that the rates and severity of weight loss differed between different tumor types. In particular, pancreas and upper GI cancer, along with lung, were significantly affected, and that this weight loss resulted in the patients having shorter survival and worsened chemotherapy response. Most of our studies, and some of the studies that I'm going to detail in the rest of the talk, really focus on patients with pancreas and upper GI cancer. So what is cancer cachexia? What are the signs and symptoms that we see in patients? Predominantly, we notice the weight loss with a real focus on muscle wasting. But patients also describe anorexia, a loss of appetite, and fatigue. Clinically, in advanced cases, we can see other things such as edema or the collection of fluid and anemia. Importantly, what are the clinical consequences of cancer cachexia? Well, first of all, patients report decreased physical functioning, decreased physical activity, and the ability to look after themselves. In turn, they demonstrate worsened response to standard chemotherapy agents and actually have higher rates of complications. Overall, these things transpire into a worsened global quality of life for the patient and, unfortunately, a shortened survival. So what is the pathophysiology of cancer cachexia? As I've mentioned previously, it appears to be the response between an abnormal interaction between the host and the tumor. The tumor is responsible for the secretion or the development of various mediators within the host, and those can be inflammatory or neuroendocrine, and the tumor itself may release some specific mediators of its own that lead to muscle wasting. The host metabolic response is also impacted upon by further things, such as the treatment that they might receive. Now, this was the initial model that was put forward, but actually one of my highest impact publication has been something that we published back in 2007, which was really where we proposed a different model. And in it, we would argue that there is no real pure model of cancer cachexia. Most patients with cancer have a range of different muscle wasting phenomena taking place at the same time. They have the tumor, but they may also be going chemotherapy, and chemotherapy can cause muscle wasting. They may have comorbidities such as diabetes. They may be sedentary or have a poor diet. And importantly, many cancer patients are elderly, and therefore they tend to have age-related sarcopenia. All of these things combine together to create muscle wasting in the individual. If we go back to the tumor-host interaction at the moment, I'm going to describe some of the work that we've done in each of these areas. So firstly, inflammatory mediators. We know that the tumor causes the development of pro-inflammatory cytokines, and that these have a number of peripheral effects within the host. They act centrally on the brain to cause anorexia. They act peripherally on the skeletal muscle to cause wasting. And they act on the liver to cause the acute phase protein response. And all of these things combined result in increased urinary nitrogen loss. Now, that was the original diagram when I got into this field a few years ago, but the diagram nowadays probably looks something more like this. And actually, there are probably far more lines, there should be far more lines on it than I've actually drawn. We know now that inter-organ crosstalk plays a significant role in the regulation of peripheral tissues. For example, muscle and fat have a lot of inter-organ crosstalk, as do muscle, fat, and bone. We know that the skeletal muscle isn't just wasted by cachexia, but also cardiac muscle. And we still have really undescribed effects from the patient microbiome and how this might affect peripheral skeletal muscle wasting. Both the brain and the liver are probably underappreciated in terms of the amount of impact and role they play within cancer cachexia. If we consider inflammation a little more globally in the concept of systemic inflammation, this was some old study data from our unit some 25 years ago, just simply looking at the survival of patients with unresectable pancreatic cancer stratified for the acute phase protein response, usually evidenced by a CRP or C-reactive protein level greater than 10. And you can see that patients who've got an acute phase protein response because of systemic inflammation have a significantly decreased survival. Perhaps more importantly than just survival alone, these patients also are anorectic and therefore are likely to have a worsened quality of life. And if you do indirect calorimetry on these patients, you show that these are also the individuals who have a higher resting energy expenditure and a hypermetabolic. One of the first studies that I was involved in cancer cachexia was involving macrophage inhibitory cytokine 1, MYC1, otherwise known nowadays as GDF15. MYC1 was first identified in tumor mouse models as a likely tumor-secreted anorectic agent with central effects on the brain. We conducted one of the first studies in human cancer patients and found elevated levels in patients with esophagogastric cancer. Although we didn't find any strict correlation with nutritional status, we did find a tight relationship with both inflammatory status and overall survival. It's through evidence like this that GDF15 is now one of the key front runners in pharmaceutical trials as a mechanism to try and reverse cachexia through GDF15 antagonism. What about neuroendocrine mediators? Our unit had previously identified abnormalities in the neurohormonal axes in cachexia, namely with reduced leptin levels or satiety signals in patients with advanced cancer. In comparison, I performed an analysis of sex hormone levels in patients, the same type of patients, those with advanced pancreatic cancer, and showed profound abnormalities in gonadotrophin levels and significant elevations in sex hormone binding globulin compared with controls. I'll discuss these findings a little further when we consider the role and impact of sexual dimorphism within cancer cachexia. Our previous work has also considered tumor-derived mediators of cancer cachexia too. One putative mediator of skeletal muscle wasting in mice is proteolysis inducing factor, a protein that is differentially transcribed and translated from the gene encoding for dermcidin, which is an antimicrobial sweat peptide. The existence of human PIF has always remained a matter of controversy. we conducted a series of studies on PIF, and in particular, we found a number of different things. We found a varied expression of dermcidin in primary human tumors explanted from patients, and also in human cell lines treated with hypoxia or oxidative stress, such as you might find within a tumor itself. However, the human PIF-core peptide detected in these samples did not demonstrate any glycosylation site, which was critical for the proteolytic activity reported by murine PIF. Furthermore, when we used the reported antibody to PIF within humans, we found that it created a lot of non-specific binding to both albumin and immunoglobulin, leading to a lot of false positives, meaning that a lot of previous studies in this area had actually been incorrect. We thought this might be the end of human PIF, but once again, I'll come back to this theme a little later on. So what are the mechanisms of human muscle wasting within cancer cachexia? Well, for protein regulation in skeletal muscle, synthesis usually roughly equals degradation with a turnover of about 1 or 2% per day. There are pathways that lead to muscle hypertrophy through enhanced protein synthesis. These pathways include things like IGF-1 going through PI3 kinase, AKT, mTOR. And then there are pathways that lead to muscle atrophy through enhanced protein degradation. These include pro-inflammatory cytokines acting through NF-kappa B. And ultimately, cachexia is thought to occur through an imbalance in these two different types of pathway. Since drawing this image approximately 14 years ago, we now have a better appreciation of other potential mechanisms that might be taking place. For example, myostatin, an autoimmune negative regulator of muscle growth, might suppress satellite cells, which are muscle stem cell activation, and thus suppress muscle renewal. There are various end effector mechanisms of muscle proteolysis. The one that's received really the most attention up to date is the ubiquitin proteasome pathway, predominantly based on some important mouse studies within the area. However, in our human studies, we've always struggled to show increased UPP activation in cancer, thus stressing that there are some important species differences in cancer cachexia and that we can't always rely on the findings of preclinical models. I'll come back to this point a little bit later on. Autophagy is now receiving increasing attention with regards to an end effector mechanism of muscle degradation. So one of the things that we've spent quite a lot of time doing in our laboratory and over the years is really examining the phenotype of human muscle within cancer cachexia. No one had really performed electron microscopy of cachectic human muscle before. And when we did this, we showed that cachectic muscle contained an increased number of fat droplets within it, a form of myosteatosis. Interestingly, the mean lipid droplet count correlated with the severity of the patient weight loss. And it also correlated negatively with CT-derived measures of visceral adiposity, suggesting that fat was being mobilized in one place and deposited in another. I'll come back to the relationship between fat and muscle a bit later on as well. We also performed other phenotypic muscle studies within our group, and these studies go to show the importance of the definition of cancer cachexia within the identification and treatment of patients. These are some cross-sectional images of rectus abdominis muscle stained for myosin heavy chain 1 and 2 alpha. And you can see that cancer patients with greater than 10% weight loss really don't demonstrate many significant differences compared to healthy controls. It's only when you get to cancer patients that have both weight loss and low muscularity, as evidenced in this case by patients identified as having low muscularity on CT, that we really see that they have a reduced fiber cross-sectional area. So these diagnostic difficulties in cancer cachexia can have significant outcomes, both in terms of patient overall phenotype and function, but also in terms of the types of treatment that we might need to initiate. We've performed multiple transcriptomic studies on muscle samples taken from patients undergoing surgery for cancer, usually rectus abdominis muscle, but sometimes combined with percutaneous quadriceps muscle biopsies. This is one such study where we took muscle samples at the time of surgery and then followed them up 8 months later after they'd had surgery with a further muscle biopsy. We showed that at baseline, cancer patients had 941 genes regulated in comparison with healthy control. These regulated genes were both anabolic and catabolic in nature, and that interestingly, prior to previous belief, downregulation of genes was more important than upregulation. At the follow-up samples, only 2 genes were still regulated compared with healthy control, showing very nicely that removal of the cancer removed the cachectic signal from the muscle. But importantly, at no point during the study did we see any regulation of the E3 ligases, or which are members of the ubiquitin proteasome pathway, just going to show that there are significant differences between mouse and human. We performed other studies on patients undergoing surgery as well. Mouse studies had shown that cachectic muscle is quite porous and that there may be some membrane abnormalities. So we looked at that as part of a collaborative study in patients with upper GI cancer and showed that those individuals had significant deregulation of what's known as the dystrophin glycoprotein complex, which is a large multi-protein complex that sits in the muscle membrane and prevents it from contraction-induced injuries. In particular, cancer patients had reduced levels of dystrophin and increased glycosylation of both beta-dystroglycan and beta-sarcoglycan. This was really the first study to show an association between acquired forms of muscle wasting, such as cachexia, and a genetic form of muscle wasting, such as muscular dystrophy. I'll show some further studies with DGC a bit later on. Having identified problems at the muscle membrane, we were also interested in what happens at the nerve-muscle interface. We therefore performed a morphometric analysis of neuromuscular junctions across control patients, weight-stable cancer patients, and cachectic patients. And contrary to the findings that are seen in age-related sarcopenia and mouse studies, we found no differences between the 3 groups, implying that the focus of pathology in cancer cachexia really is the muscle itself, nothing proximal to that. And it also suggests that electrical stimulation may still be a valid form of potential treatment for cancer cachexia. Initially, most research in the cachexia field was based in muscle, largely because muscle was thought to be the tissue that most regulated patient function and therefore quality of life. However, studies began to emerge which showed that muscle may actually be regulated, or muscle loss may be regulated through fat muscle crosstalk. For example, there was one seminal publication in Science in 2011 which showed that mice that were knockout for adipose triglyceride lipase were essentially resistant to muscle loss when implanted with a tumor, which led to the theory that it was the resistance to lipolysis that was preventing the muscle loss. So we began to investigate fat wasting. The These show some 71 patients with esophagogastric cancer who have had CT body composition analysis across neoadjuvant chemotherapy. We showed that it was female patients and elderly patients that really had the highest rates of adipose loss in this area, particularly in relation to subcutaneous adipose tissue. We also performed microarray transcriptomics, and we showed that there was a significantly greater number of regulated genes in visceral adipose tissue taken from the greater omentum compared with subcutaneous adipose tissue, and that this regulation was enhanced in patients with cachexia, or as on this table, CWL, which stands for cancer weight loss. Genes involved in inflammation were enhanced in visceral versus subcutaneous adipose tissue comparisons in both control and cancer patient cohorts, but there were different gene sets in each type of patient, which is quite an interesting concept to consider. The gene showing the largest difference in expression was ITLN1, a gene encoding for interleukin-1, which is a novel adipose cytokine, which has been investigated previously in other causes of weight loss, but has never been identified previously in cancer cachexia. So this was the first description. Energy metabolism and adipogenesis genes were downregulated in cancer visceral adipose tissue, but interestingly, so were the genes involved in fat browning. Once again, this is a difference between mouse and human cachexia, since previous murine studies had demonstrated that parathyroid hormone-related protein might be a regulator of fat browning in cachexia. We wanted to investigate interleukin-1 a little bit more, and we showed that when we used ELISA to assess tissue concentrations, these were definitely elevated within the cancer cachexia population. However, we didn't see the same findings within the circulating plasma from the same individuals, suggesting that interleukin-1 might be more of an autocrine signal located within the fat itself. And we're conducting further studies in this area. So the findings of our fat wasting studies were really that adipose tissue is clinically relevant in cancer and cachexia, and that both subcutaneous adipose tissue and visceral adipose tissue have unique gene expression signatures in cancer. Visceral tissue is metabolically active and might be a target for therapeutic manipulation in the future, particularly if it's a gateway for skeletal muscle loss. But the downregulation of any energy metabolism implies a limited role for fat browning in humans compared with mice. One of the areas in which we've done a lot of work in the past and feeds on from some of the muscle phenotype studies that I was showing earlier is that we're really interested in what patients get cachexia and how we identify them ahead of time. Now, that might be in terms of risk factors so that we can consider treatment for those individuals, but it also feeds into the clinical trial platforms and what inclusion criteria that we should use. One of the areas that we were initially interested in was sexual dimorphism. This was a phenotypic study using measures of power and strength, and we showed that quad strength, lower limb power, muscle cross-sectional area, and muscle quality were reduced in male patients, but only strength and quality were reduced in female patients. Physical function and fatigue were reduced in males again, but not in females, suggesting that impairment of muscle mass quality and function and how that impacts on the quality of life of the individual varies not only with weight loss, but also sex of the patient. So that's looking at a kind of clinical phenotypic level, but we also saw changes at a biochemical level. As I referred to earlier, we did some neuroendocrine studies on our patients. And we showed that actually in individuals with advanced pancreatic cancer, approximately, well, almost 3/4 of males are hypogonadal when you measure calculated free testosterone. Hypogonadal males demonstrate reduced BMI and also reduced hand grip strength. And importantly, and unfortunately, these individuals also appear to have significantly reduced survival. Now, what we don't know is quite whether or not this is due to the impact on the muscle or whether or not this is due to a relationship with systemic inflammation, because it's also the hypogonadal patients who have the highest rates of inflammation. In females, we see an opposite pattern. Almost 1/5 of postmenopausal advanced pancreatic patients who are female demonstrate hyperoestrogenal or hyperoestrogenism. And once again, it is these individuals who are pro-inflammatory and have worsened survival. We know that opioid use is a major cause of iatrogenic hypogonadism, particularly within males. So it seems that based on these studies that we showed that opioid use is also associated with worsened survival. So one of the drugs that we prescribe routinely and regularly for the palliation of pain associated with advanced cancer may also have the unexpected side effect of hastening death. So that was sexual dimorphism, but do patients have a genetic predisposition to cancer cachexia? Having identified sexual dimorphism in patients, we performed some large European palliative care research collaborative studies, and in particular, we performed candidate gene association studies and found that one allele of a single nucleotide polymorphism in the gene encoding for p-selectin, a cell adhesion molecule involved in platelet activation, was associated with cancer-related weight loss. Synchronous mouse studies also suggested a role for p-selectin in systemic inflammation when mice were injected with bacterial lipopolysaccharide. We repeated these studies in other large cohorts, and we combined the weight loss data with CT body composition analysis to obtain more strict criteria for cachexia. And we showed that the combination of weight loss and low skeletal muscle index associated with polymorphisms in the leptin receptor, the myostatin receptor, TNF, and angiotensin converting enzyme, all plausible mechanisms for cancer cachexia. Other things that we've considered in patient selection is screening. The global leadership in malnutrition authors, you may have heard of the GLIM criteria, have suggested that all patients at risk of malnutrition should undergo a two-stage process of identification. The first stage involves screening, and then the second stage involves assessment, diagnosis, and severity classification. We therefore performed a comprehensive psychometric assessment of the various validated screening tools available for cachexia, sarcopenia, and malnutrition to try and see if we could identify a screening tool that could be used universally for all three different types of condition. We showed that only one tool was really validated for cachexia, and that's known as the Casco tool, and that no single screening tool could be implemented for the simultaneous assessment of all three conditions. This led us to hypothesize the constituents of an ideal screening tool, and we thought that those should include quantification of weight loss, measurement of patient BMI, assessment of dietary intake, the underlying health state as to whether or not the patient has an underlying chronic disease, age, a direct assessment of muscle mass and function, and also any measure of metabolic derangement such as systemic inflammation. If we take it to the second stage of the GLIM process, well, the field of cachexia really moved on in 2011 when the consensus definition of cachexia was published. And this showed that, or defined cachexia as a multifactorial syndrome characterized by an ongoing loss of skeletal muscle mass with or without loss of fat mass that cannot be fully reversed by conventional nutritional support, and which leads to progressive functional impairment. The pathophysiology is characterized by a negative protein and energy balance driven by a variable combination of reduced food intake and abnormal metabolism. And the diagnostic criteria was stipulated as weight loss greater than 5% or weight loss greater than 2% in individuals who already had a low BMI or were sarcopenic by CT criteria. The authors also described cachexia as a journey, 3 stages through which a patient may pass. Pre-cachexia, prior to significant weight loss, cachexia as the syndrome that we might all know, and then refractory cachexia, where the patient is essentially close to death, has a low performance score, and any treatment might be ethically and feasibly impossible. Our unit have always been interested in using CT body composition analysis, and we've shown that not only is sarcopenia a problem, but the combination of obesity and sarcopenia is a really bad combination, and that results in a significant reduction in patient survival, but also it's been shown in other studies to be associated with things like post-operative morbidity in surgical patients. Because of this prognostic value of CT body composition analysis, it has been proposed as a method of or a valid outcome measure for use in clinical trials to use it to demonstrate increased muscle mass following treatment. So we really wanted to analyze its validity as an outcome measure, and using published cutoffs for muscularity, we noticed that there were high rates of apparent sarcopenia, shown here in red, even in healthy control patients who were undergoing live kidney donation. We also showed sarcopenia in vascular patients undergoing aortic surgery and cancer patients, as you might expect. The implication is that one safe threshold for skeletal muscle increases when one is diagnosed with cancer, which is why you can be apparently sarcopenic and still healthy at the same time. We wanted to look also at the relationship between skeletal muscle index as demonstrated by CT with other relevant outcomes that you might expect to improve following intervention, including muscle protein content, muscle cross-sectional area, patient functional status, and cancer patient quality of life. And unfortunately, CT body composition analysis didn't really correlate significantly with any of them. You can see here a low relationship between muscle index on CT and protein content, suggesting that CT body composition analysis is prognostic at baseline, but it may not be a sensitive outcome measure for clinical trials. This finding also led us to speculate further that there may not be a linear relationship between muscle mass as we can record it and physical function, an assumption that has always been made previously in clinical trials. We hypothesized that actually the relationship might be curvilinear, and that the same trajectory may not be followed during muscle recovery as during muscle degradation. And we have to consider these things when we design our clinical trials and when we choose our relevant outcome measures. What about biomarker programs? Well, as I talked about briefly, we have considered things like dystrophin glycoprotein complex in our previous studies, and we've shown therefore that muscle samples taken at the time of surgery with curative intent can actually predict patient long-term survival. So this is a sample that's been taken early on in patient treatment, and yet it has had a profound discovery with regards to patient long-term prognosis. And this is why we emphasize the need in the future that when it comes to cancer cachexia, we shouldn't just stage the cancer, we also need to stage the host. We've shown the same with myosin heavy chain levels taken from muscle samples during surgery. We've used mass spectrometry to look at the urine of cancer patients. And again, we've shown signals within urine that can detect patients with significant weight loss. In this particular study, you'll notice that the two candidate biomarkers that we identified were myosin 2, as you might expect from muscle degradation, but also, interestingly, dermcidin, the progenitor molecule for PIF, and this is something that we need to explore further. We've done similar studies using plasma rather than urine, but this time focused on metabolomics, and we've identified metabolic profiles that are able to identify patient weight loss with very strict accuracy. With regards to cachexia management and clinical trials, we've been involved in a number of different trials in the past, and we've very much moved to a system now where we want to focus on multimodal trials. The top trial there details formoterol, which is a beta-2 agonist associated with an appetite stimulant, midgestrol. And this was able to demonstrate an improvement in muscle volume within the individual patient in phase 1 studies. I'm currently involved with some post-hoc analysis of the anomorelin trials. Anomorelin is a ghrelin agonist, and we've just received funding for a new anti-interleukin-1 alpha trial from the MRC. This is just to expand on our concept of multimodal trials. Any particular trial really has to include various things. We need to provide the patient with nutrition, but we also need to provide a pharmaceutical intervention, which is anabolic, probably anti-inflammatories, combine that with exercise where possible, and optimize standard anti-neoplastic treatment at the same time. We'll talk briefly about some outcome measured development that we've done in the past with regards to CT scan, but one area I'll just touch on briefly is also the work that we've done with regards to physical activity. The physical activity level of the patient is largely governed between the balance between total energy expenditure and resting energy expenditure. We know that patients are often hypermetabolic, but actually, interestingly, TEE is often not that altered. It's because of a reduction in physical activity level that the TEE remains the same. We've shown previously that TEE can be improved through an anti-inflammatory treatment supplied to patients, and in this case it was icosapentaenoic acid fish oil based supplements. Using developments in wearable technology, we've been trying to develop physical activity as an outcome measure. In this case, this particular study involved accelerometers attached to the patient's thighs. And we showed that in patients undergoing chemotherapy for cancer, they spent 2 hours per day less upright, and approximately 40% had a 40% lower step count compared to age-related controls who were attending a bowling club. So in conclusion, ladies and gentlemen, I've tried to go through a lot of ground in this talk, and I've talked about the various areas that we've been involved in. I've discussed mediators, some of which have shown up positive, some which PIF and NMJs, which may turn out to be less important. I've discussed patient selection and talked about the roles of sexual dimorphism and genetic predisposition, and also different methods by which we can identify patients both in clinic and in clinical trials. I've briefly touched on some of our RCTs, and we're moving forwards in this direction currently. And lastly, at the end, just briefly discussed some issues regarding physical activity. So what's the future for cancer cachexia? It's actually a very busy time at the moment. Consensus groups have been formed with regards to redoing the definition of cachexia, considering what endpoints really are best for patient analysis, and also how we should educate clinicians about cachexia. People may have noticed that the CI UK NCI Grand Challenge has just been released about cancer cachexia, and this is really a turning point for us, since large-scale funding like this has largely been absent in the past. And as discussed very briefly at the beginning, we've seen the inception of some new and important collaborations, such as the Cancer Cachexia Society and the NIHR Cancer Nutrition collaboration. I'd like to thank all of the team in Edinburgh, and in particular, Professor Kenneth Fearon, who unfortunately passed away a few years ago. He was really the key man in all of this work and was my MD supervisor. He was a world-leading expert in the field and has really pushed things forward. He is sadly missed. I'd also like to thank all our collaborators, particularly the ones that have detailed in these studies across the USA, Canada, Europe, and Australia. I'd like to thank all of our research partners and funders. And in particular, I'd like to thank the Nutrition Society for the opportunity to speak today and for this award, for which I am very grateful. Thank you very much.
Well, that was absolutely splendid, Richard, and thank you for such an all-consuming and all-encompassing presentation. And I know Professor Fearon and Sir David Cuthbertson would be shining down on that with great pride, you know, as to what you've done. And so thank you very, very much for the delivery. Now, I'm sorry I can't, together with the NUTSOP team, open up all the delegates to give you a clap, but I will give it on behalf of everybody listening. And I know we have great numbers joining us, as indeed our Lady President, Professor Lovegrove, said at her beautiful welcoming introduction earlier on today. So now we would like to present you with your award. Now, I'm sorry, my hand won't stretch that far from Surrey to Edinburgh to give you a handshake, Richard, but I know that you have your award on you. So I wonder if you wouldn't mind just holding it up for everybody to see. So this is the Cuthbertson Award, which is a bit special. And Richard, you really, really were a most, I said that at the beginning, didn't I? And by the time we've got to the end, you've proven it absolutely, that you really are the worthy winner. And of course, it is a bit special because it's the 30th year of the award. So Richard, congratulations from us all, from the Nutrition Society team, Lady President, honorary officers, and indeed all the ladies and gentlemen who are joining us, you know, across the world, listening to that wonderful, wonderful presentation. So Richard,
thank you very much. That's really kind.
You are very, very welcome. Well, I, as is with all the awards, there are no questions, so we won't give you a grilling. As I was listening through, I was thinking, gosh, there's so many things we could ask you, but that's always what we do with the awards. We don't give you questions, but just to say thank you very much on behalf of everybody for that, you know, beautifully presented presentation. And we are now going to close this of the third awards, but we encourage you all please to come back for 3 o'clock for the final one, which is of course the Elsie Widdowson Award, which will be chaired by Professor Paul Traherne, and of course will be to Professor Anne Prentice OBE. And so that will also be as equally special. So with no further ado, congratulations, Richard, and good afternoon to everybody.
Good afternoon.